Hello everybody, thank you so much for your time. My name is Zuhair Khan and today I will be discussing an economical protocol for neuroanatomy instructors and researchers, combining the Nissl and NADPH-diaphorase protocols to overcome their individual limitations. I would like to first begin by providing a brief background on the techniques combined to develop today's protocol. You can think of these techniques as colors on a palette that are used to paint specific biological targets. Nissl, being the red in today's images, stains ribosomal RNA, which is found in every cell's cytoplasm. This allows for consistent labeling of whole neuronal populations, which can be seen here in the bottom right of a human hippocampus slice that has been stained with the Nissl dye neutral red. Each red dot is a stained cell, and because all of the cells are stained, it makes it easier to determine what structures are present. For example, like this organization here, this is clearly the dentate gyrus of the hippocampus. The disadvantage to the Nissl stain is the limited information regarding neuronal morphology, and as you can see, there aren't really any visible processes like axons or dendrites visible. This is where the blue NADPH-diaphorase stain comes into play. It stains discrete populations of neuronal nitric oxide synthesizing interneurons and shows these specific interneurons in their entirety, meaning that you can see their axons and dendrites. The only issue here is that we don't really know where these stained interneurons are located relative to other structures. And this is where the big question comes in. What are the benefits of combining them, and how does this allow them to overcome their individual limitations? So I'll sprinkle a little bit of Nissl and a little bit of NADPH-diaphorase, and let's see what we get. Wow. One of the stain's best features, I must say, would be the fact that it is beautiful, and I mean truly gorgeous, breathtakingly beautiful. You can see these gorgeous cell layers running through the hippocampus, allowing for identification of what structures are present, and you also have these darkly stained NADPH-diaphorase interneurons that you can use to observe where they are located and next to what structures. The slice can now provide us with so much more information, and I will go over its applications later in the presentation. Here is the reaction for the NADPH-diaphorase histochemical reaction. This end product, nitroblue tetrazolium formazole, is the dark blue you see in the stained neurons. It is soluble, meaning that if you expose it to the Nissl dye, which is almost all water, it will dissolve. As a result, some tinkering had to be done in order to develop the counterstain. You can see that when you expose the NADPH-diaphorase stained slices to the Nissl dye for 2 minutes, sufficient Nissl staining doesn't occur. On the contrary, when you expose it for 5 minutes, the Nissl dye washes away most of the NADPH-diaphorase staining and then ultimately overpowers the stain. 3.5 minutes was seen to be a good optimal time that created a good balance between the two. The protocol is relatively straightforward. You have your cut sections that are incubated in the NADPH-diaphorase medium. You then dehydrate and defat your tissue. Now, because the Nissl dye is hydrophilic, it needs water, so you must rehydrate the tissue, but this time using quick rinsing, meaning that you only submerge it in the solvents for 1 to 2 seconds rather than the normal 3 to 5 minutes. You then stain it in the Nissl dye for 3 to 5 minutes, and now you must once again re-dehydrate the tissue using the new quick rinsing method, and once you're back up into the defatting solvent, which is the xylene, you can cover slip your slides. And the results produced are quite nice, if I do say so myself. As you can see here, with just NADPH-diaphorase staining, the granule cell layer is completely devoid of staining, and you don't really know what exact layer these neurons are in. With the counterstain, however, it now becomes relatively clear where these neurons are located. These ones here are clearly in the subgranular zone, whereas this neuron right here is most definitely in the granule cell layer. A similar benefit happens in the olfactory bulb, where prior to counterstaining, it seems kind of devoid and lifeless, but with the counterstain, you now have many more layers that become present, such as the mitral cell layer right here. Additionally, you can now see the relationship of these periglomerular cells that are located in the dorsal and medial parts of the olfactory glomeruli. In the rat kidney, with just NADPH-diaphorase staining, there's not much that can be seen or observed. But with the counter stain, you can see the crystal violet stain tissues here on the left and the neutral red stain tissues here on the right. There are much more structures that are observable, such as the vasa recta, right here with its red blood cells, the kidney glomeruli, and the macula densa. This image here of the salivary gland has been shown as an example as to why we use the neutral red dye instead of the crystal violet. The crystal violet makes it difficult to distinguish between the NADPH-diaphorase stain and the crystal violet Nissl dye. The red and blue with the neutral red dye is much more convenient and optimal to use. I will now go over the applications that instructors and researchers can use. So, instructors such as Dr. Kageyama can use this image to draw the network and anatomy of the hippocampus and use it for teaching purposes. Researchers, such as myself, use it to divide specific regions of the brain, such as the hippocampus, into its specific layers and then see how the number of stained interneurons changes based on exposure to enriched environments. If that sounds interesting to you, please check out my other presentation at a different session in this conference. And lastly, one of the biggest advantages to the counterstain is that it is cost efficient and convenient. The total cost of the counterstain reagents comes out to about $335, whereas if you use other conventional methods such as immunofluorescence to look at the same things, that cost is about $605. Additionally, the counterstain is much more stable, doesn't require many controls, and is not susceptible to photo bleaching. Overall, in today's protocol can be visualized by looking at this simple icon here in the middle. The counterstain combines the neutral red dye, which stains whole neuronal populations, and the NADPH-diaphorase stain, which stains discrete interneurons. This combination and overlapping of the two allows them to overcome their individual limitations and provides us with a gorgeous end product that allows for extensive scrutiny of tissue. Due to its cost efficiency, convenience, and efficacy, it is a great tool to be used by both instructors and researchers. I would like to thank everyone here who had some part in preparing the tissues and or slides or was someone I taught this technique to. Thank you so much for the Teacher Scholar Grant of 2018 to 2019, and thank you so much, Dr. Kageyama, for everything that you do. I'm now open up to the Q&A session, and I am very grateful for everyone who listened, and thank you so much for your time, and I really hope that you enjoy this presentation. If you have any questions or would like a more in-depth, list of the steps in the protocol, please contact me.